Hi everyone. In this session I'm going to talk about the blood groups and blood types. A blood type is a classification of the blood based on the presence or absence of different antigens that are expressed on the surface of the red blood cells. And these antigens belong to at least 43 different blood group systems that were identified so far in the human being. And when we talk about the blood type, we are describing the combination of these antigens that belong to these different 43 blood group systems. The two most important blood group systems are the ABO and the RH systems, and they determine whether someone's blood type is suitable for blood transfusion or not, to make sure that transfusion reaction won't happen if someone needs blood. In addition to the very common blood group systems that are ABO and the RH systems, we have other systems. As I mentioned, we have a total of 43 blood group systems, and for example, we have the MNS system, we have the KELL system, and we have the Lewis system. But we focus mainly on the ABO and the RH blood group systems because these are the antigens and antibodies of these systems are responsible for most of the transfusion reactions that happen in the clinic. So the objectives of this session are to talk about the different blood group systems, and the two main systems are ABO and the RH systems. Explain why it is so important to match donor and recipient blood types before the administration of blood transfusion. Then I will talk about the transfusion reactions and hemolytic disease of newborn. And then I will talk about blood typing and cross-matching, how to know the blood type of a person and what is meant by cross-matching. So let's start with the ABO blood group system. The ABO blood group consists of, as I said, it is determined by the presence of antigens or surface antigens that are expressed or present on the or are part of the lipid or the phospholipid bilayer of the red blood cell membrane. So we have two antigens. We have the A antigen and we have the B antigen. These antigens, if they are present or absent, then we can know which type we have based on the ABO blood group. So we have two antigens, A and B, on the surface of the red blood cells. We have type A blood. When we have type A blood, it means that A antigen is present on the red blood cells and only A antigen. If the person has type B blood, then it means that he has only the B antigen on the surface of his red blood cells. If the person has the AB type blood, then it means that he has the A and B antigens. Both of them are present on his red blood cell membrane. Then if a person has a type O blood, it means that none of the antigens are present on his red blood cell surface. We have antigens, it means that also we have antibodies. So in this ABO blood group, we have two antibodies, the anti-A antibody and the anti-B antibody in the plasma. And we also call them agglutinins because if they interact with red blood cells that express the respective antigen, then they will cause their agglutination. They will clump with each other. They will not exist as free red blood cells anymore. So if a person has a type A blood, then he has the anti-B antibodies in his plasma. And if anti-B is present, it won't react with red blood cells that express the A antigen, and this is quite logic. On the other hand, if the person has type B blood, then he has the anti-A antibodies. Anti-A antibodies won't react with the B antigen on the surface of the red blood cells. A person with type AB blood has no antibodies because the presence of any of these antibodies can cause the agglutination of the red blood cells and their destruction, and we don't need that. And then finally, the type O blood has both the anti-A and anti-B antibodies in his plasma. These antibodies won't find any antigen on the surface of the red blood cells of type O blood person, because type O means that the red blood cells do not express any of these two antigens. So in this image we can see clearly we have the blood type, we have type A, type B, type AB, and type O. Then we have the red blood cells and we have the plasma. So for the type A person, we have A antigen on the surface of his red blood cells, and we have the anti-B antibody in his plasma. They won't react, so we have no agglutination. We don't have any reaction and no hemolysis, no destruction of red blood cells. For type B person, there are B antigens on the surface of red blood cells and anti-A antibody in the plasma, and this is safe. No reaction will happen and nothing will occur. Then a person with type AB, he has both A and B antigens on the surface, but none of the antibodies in his plasma. And finally, for the type O person, we don't have A or B antigens on the surface, so no antigens on the surface of red blood cells. On the other hand, we have the anti-A and the anti-B antibodies in the plasma or in his plasma. So a summary here of the ABO blood group interactions. So we have the blood type. When we have the A type, the antigens that are present on the red blood cells are the A antigens, and the antibodies in the plasma are anti-B antibodies. For the type B, then we have the B antigen on the surface of red blood cells and anti-A antibodies in his plasma. For type AB, we have both A and B antigens on the red blood cell surface, none of the antibodies in his plasma. For the O type, none of the antigens on the surface of red blood cells, but we have both anti-A and anti-B antibodies in the plasma. About the compatibility or the compatible donor and incompatible donor, I will talk about that in one of the coming slides. Usually we call the blood group O as universal donor, and blood group AB is called universal recipient, and I will tell you why in one of the coming slides. Now let's talk about the Rh blood group. What are the antigens here and what are the antibodies? Rh blood group, it is named because the antigen was first discovered in the blood of the rhesus monkeys. That's why we just get the abbreviation from the first two letters of this type of monkeys. The Rh blood group consists of only one antigen, so things here are easier, and we call this antigen that is expressed on the surface of red blood cells the Rh antigen or the D antigen. So in some textbooks it is called Rh, in other textbooks it is called D antigen, and they have the same meaning. A person with type Rh positive blood has the Rh or the D antigen on the surface of his red blood cells, while a person with Rh negative blood type has no Rh antigen on the surface of red blood cells. And another interesting and important fact about the Rh blood group, that we don't have antibodies or anti-Rh antibodies in the plasma. So we don't have pre-formed antibodies. The plasma normally does not contain anti-Rh antibodies, even in Rh negative blood type. So no preformed antibodies, which is opposite to the ABO blood group. In the previous slides, I explained that always we have the antibody to the missing antigen in the plasma of the person. But here, even if the person has no Rh antigen on his red blood cell surface, he has no anti-Rh antibodies in his plasma. So there are no preformed antibodies in the plasma, even in the Rh negative blood type people. If an Rh positive blood is transfused to an Rh negative person, then the immune system will form anti-Rh antibodies. This is the only case where anti-Rh antibodies are formed. And these anti-Rh antibodies will remain in the plasma of the recipient, and if he receives a second transfusion of Rh positive blood type, then agglutination and hemolysis will happen. Why? Because now he has anti-Rh antibodies in his plasma, and once he receives another transfusion of Rh positive blood, the anti-Rh antibody will interact with the antigen or the Rh antigen on the donated red blood cells, and this will cause agglutination and hemolysis of the donated blood. So this is one of the major problems with the Rh blood group. It is very common to hear that certain blood types are common, other blood types are rare, and we have the conception that usually the O blood type is very rare. This is not the case. As you see here we can see the distribution of blood types in the USA, and similar figures happen in other countries all over the world. It is not the O type that is rare, because as you can see, it is almost 50% of all the blood types in the population, and even more than that. What is rare is the Rh negative. As you can see that the majority of the population are Rh positive, and some ethnic groups, they don't have Rh negative type. They are 100% Rh positive. So the rare thing is the existence of Rh negative people. So if we say, for example, O positive blood type, it is not rare, but the O negative is really rare. And if a person with O negative blood type needs transfusion, then he will suffer to find a suitable donor. Now let's talk about the transfusions, because the main reason for knowing the different blood groups or the different blood group systems and the blood type of a person is transfusion, blood transfusion. When a person needs blood, he should get blood from the donor, and before giving the blood from the donor to the recipient, we should make sure that no transfusion reaction will happen, because if there is incompatible blood transfusion, the red blood cells or the antibodies in the plasma will interact with antigens on the surface of red blood cells. This will cause agglutination of the red blood cells, and the agglutination will block small blood vessels, and they stop or block the blood flow to the tissues, so the tissues will suffer from ischemia. And another problem, that agglutinated red blood cells will undergo hemolysis. Hemolyzed red blood cells will release their content of hemoglobin. Free hemoglobin cannot carry oxygen. Hemoglobin is able to carry oxygen when it is present inside intact red blood cells. Free hemoglobin has no ability to carry oxygen from the lungs to the peripheral tissue. Another problem with hemoglobin, that it will go to the kidneys and will precipitate there and cause renal failure. So this is another major issue with hemolysis. So transfusion is the transfer of whole blood or blood component. We can just fractionate the blood. For example, we take packed red blood cells, platelet concentrate, we take the plasma, we can take the antibodies, and so on. So it is the transfer of whole blood or blood components like red blood cells only or plasma only of one person, we call him or her the donor, into the bloodstream or directly into the red bone marrow of another person, and in this case he or she is called the recipient. The transfusion is most often given to alleviate anemia as a treatment of anemia, or to increase the blood volume after severe bleeding or hemorrhage, or to improve the immunity because the blood contains immune cells, contains antibodies, and this would help in improving or increasing the immune response. Donated blood can be separated into various components to make better use of it. Whole blood can be fractionated into units of packed red blood cells, so those people who need only red blood cells can be given only red blood cells. We can get also fresh frozen plasma, and this is very rich in plasma proteins, including antibodies. We can also isolate the platelets. We can have platelet concentrate, and also we can get the white blood cells that are very important for the immune response. And finally, we can get different proteins like albumin, coagulation factors, and antibodies can be individually collected, so separated from each other and given to the appropriate patient as a treatment. So we can use one unit to get or to treat several patients based on their needs. So how to know whether there is blood type compatibility before starting the the blood transfusion process? It is very common to see such an image in a textbook, and it is quite complicated, or it is not easy to memorize. And this is only for the ABO blood group system. And if we add the RH blood group system, then it will become more complicated. But how to make it simple and easy to remember, and also easy to remember during the exam to answer questions that are related to the blood transfusion. I can show you here an easy way how to remember that in any exam and answer any question with ease. Let's start with the ABO system. The AB blood type can only give blood to a person with the same type. So AB type or donor with AB type can give blood to a recipient with AB type. A person with A type can give to a person with AB type, and also can give to a person that has A type blood. Then a person with B type can give to a person with AB type, and also to another person of the same type, to the B type. A person with O type can give to anyone, can give to the A type, can give to a person who has B type, or a person who has the AB type, and of course can give to a person that has the O type. That's why we call the O, or person with blood type O, as universal donor, as it can give to everyone, including people with O type, while the AB type is the universal recipient. It can receive from all other types and cannot give to anyone else. So this is the universal recipient. When we add to that the RH blood group system, here we have the RH positive blood type, where the red blood cells express the D antigen or the RH antigen on their surface. It can give only to another person that has the RH type. While the RH negative can give to a person who is RH positive and to a person who is RH negative. So RH negative can give to both RH negative and RH positive. So if you have any question in the exam, and you want to know whether this blood transfusion is safe or not. For example, can we give blood type AB positive to a person who is AB negative? AB positive, so we are talking about giving AB to AB. It is possible, but since positive cannot give to negative, so we cannot transfuse AB positive blood to AB negative blood, because the arrows go only in this direction, not in the opposite direction. Another example, can we give A negative blood to AB positive person or recipient? A can give to the AB, so this is possible. A negative, so negative can give to the positive, so this transfusion is safe. I hope this will help you to answer any question in any of the exams or any question related to the blood transfusion. So what happens if we give incompatible blood? We just made a mistake, or for any reason a mismatched blood type was given to the recipient. Transfusion reactions occur when mismatched blood is infused into the recipient. The donor's red blood cells are attacked by the recipient's plasma antibodies or agglutinins, and this will cause what? So here we have the recipient, the donor red blood cells, and we have the recipient antibodies. So we are giving blood, these red blood cells, and the plasma of recipient contains these antibodies. So they will interact. There will be agglutination or clumping of the red blood cells first, and this will block the small blood vessels, and later on there will be hemolysis. Destruction of the red blood cells, the release of their content, and we know very well that red blood cells are very rich in hemoglobin. So now we have hemoglobin free in the plasma. So agglutination, which is clumping of the red blood cells, this will block or impede the blood flow, especially in the small blood vessels. Then we'll have hemolysis, which is rupture of the red blood cells, release of free hemoglobin into the bloodstream. Since we have free hemoglobin, the free hemoglobin is not able to carry oxygen, so we have low oxygen carrying capacity of the blood, and the free circulating hemoglobin will precipitate in the kidneys and will cause renal failure, as I mentioned earlier. Now let's talk about Rh incompatibility, and this is a common problem, especially when an Rh positive male marries an Rh negative female. So in the pregnancy there is a risk of having this kind of Rh incompatibility, because the fetus, based on the genetics, could be Rh positive similar to his father, or Rh negative similar to his mother. If the fetus is Rh negative, then there is no problem. But if the fetus is Rh positive, this will trigger the mother immune response to produce anti-Rh antibodies, because the Rh negative mother has no preformed antibodies. This is the first exposure to the Rh antigen, and this will stimulate the mother immune response to produce anti-Rh antibodies that will attack the fetus in the next pregnancies and could lead to the hemolytic disease of newborn. So the most common problem with Rh incompatibility is the hemolytic disease of the newborn that may arise during pregnancy. Normally, there is no direct contact between the maternal and fetal blood, because we have this placental barrier while the woman is pregnant. However, small amount of fetal blood leaks into the maternal circulation during delivery. So there will be very small amount of blood, of fetal blood, that will enter the maternal circulation during the delivery process, and this will trigger the immune system of the mother. If the pregnant woman is Rh positive, then we have no problem whether the father is Rh positive or Rh negative, because no Rh incompatibility will happen. So whether the fetus is Rh positive or negative, or whether the father is Rh negative or positive. If the mother is Rh negative, then we have two possibilities. If the fetus is Rh negative, so there is no problem. Even if small amount of fetal blood will enter the circulation, the fetus red blood cells do not carry the Rh antigens, so nothing will happen. The immune system will not be stimulated. If the fetus is Rh positive, the first Rh positive fetus will not be affected, but the small amount of his blood that will leak into the maternal circulation will stimulate the mother immune system to produce anti-Rh antibodies. This anti-Rh antibody will attack the red blood cells of any future Rh positive fetus, and this will lead to the agglutination and hemolysis of the fetal red blood cells. So even if the fetus is Rh positive, the first pregnancy will go safe because the mother will be exposed to the fetal blood during delivery, and this is very short time to produce antibodies. But after the delivery, the maternal immune system will produce anti-Rh antibodies that will remain in the plasma of the mother and will attack any future Rh positive fetus. So next pregnancies of, especially with Rh positive fetus, will be at risk of hemolytic disease of newborn. So as you can see here, we have an Rh negative mother, we have the Rh positive fetus, and his red blood cells or her red blood cells express the Rh antigen. During the delivery, small amount of fetal blood will enter the maternal circulation, and this will stimulate the formation of anti-Rh antibodies. If the mother in one of her next pregnancies is pregnant with Rh positive fetus, the anti-Rh antibodies will attack the fetus and his red blood cells, and this will cause hemolytic disease of the newborn. So all Rh, how to prevent that, how to avoid having hemolytic disease of newborn, or how to avoid the formation of anti-Rh antibodies by the mother. So it is very important that all Rh negative mothers receive the RhoGAM injection, which is anti-Rh antibodies. So it is an injection that contains anti-Rh antibodies. What is the function of these antibodies? They will circulate in the maternal blood, and if they encounter any Rh positive red blood cells that came from the fetus, they will bind to the antigen. So now the antigen is covered, it is neutralized, so the immune system of the mother will not realize that this antigen is present in the mother blood. So the function or the main action of these anti-Rh antibody that is present in the RhoGAM injection is to neutralize or to cover the Rh antibody that is present on the fetal red blood cells that leaked into the maternal circulation. So it will be masked, and the maternal immune system will not confront or recognize the presence of this Rh positive red blood cells. So this injection should be given soon after delivery, because this is the time when the small amount of fetal blood enters the maternal circulation, or even after abortion or miscarriage, to avoid any exposure of the maternal immune system to Rh positive red blood cells. These anti-Rh antibodies bind to the fetal Rh positive antigen that is present on the red blood cells and will neutralize it or inactivate it. The mother immune system will not produce its own anti-Rh antibodies because the antigen was neutralized by the anti-Rh antibodies that were injected or that were present in the RhoGAM injection. And this process or this action will prevent the hemolytic disease of newborn in any next pregnancy with a Rh positive fetus. This injection should be repeated after each delivery, and it is a safe practice, especially in poor countries, is to give the injection after any delivery or miscarriage, regardless whether the father is Rh positive or negative. Because in some countries, if the mother is Rh negative and the father is Rh negative, they don't take any action, because for sure the fetus will be negative, will be Rh negative. But in some countries the typing is not correct. The father could be Rh positive, but the result shows that he is Rh negative, and this will create problem. So better to give the RhoGAM injection after any pregnancy or miscarriage. How to do the blood typing? How to know our blood type? It can be done by machines and the recent labs, but there is a very simple process of doing the blood typing. We just need to have bottles that contain solution of anti-A antibody, anti-B antibody, and also we can have the third one for the anti-Rh antibody. Then we take a small drop of blood on a glass slide, the glass slide that we use for the microscope, and then we add a drop of the antibodies, or the solution that contain the antibodies for the three different antigens: the A antigen, the B antigen, and the Rh antigen. So if the blood agglutinate with the anti-A serum, it means that the red blood cells have the A antigen, so it is the A type. And of course it won't agglutinate with the anti-B because the B antigen is missing. So if it is only, or if only the red blood cells agglutinate with the anti-A, not the anti-B, then it is blood type A. If the blood drops or the red blood cells do not agglutinate with the anti-A but agglutinate with the anti-B antibodies, it means that we have the B blood type. If the agglutination happens with both anti-sera, or the solutions that contain anti-A as well as the anti-B, then it is the blood type AB. If there is no agglutination, it is blood type O. Of course, we don't mix the anti-sera on one drop. We just get 3 drops on the glass slide. To the first drop we add anti-A and we see whether there is agglutination or not. To the second drop we add anti-B and we check whether there is agglutination or not. And to the last drop we add anti-D to know whether it is Rh positive or negative. If there is no agglutination, it means it is Rh negative. If there is agglutination with anti-D, it means the person has the Rh antigen, so his blood type is Rh positive. So when anti-serum containing anti-A or anti-B or even anti-D antibodies are added to the blood, if agglutination happens, it means that the corresponding antigen is there, and we can say that this antigen is present on the red blood surface. Agglutination indicates a positive reaction, and single drops of blood are mixed with different anti-sera. So very small drops on the glass slide, usually put 3 drops, 3 separate drops from the same person on the glass slide, and then we add the 3 anti-sera. Agglutination with any of the anti-sera indicates the presence of the respective antigen on the surface of the red blood cells of that person. So for example, if we have a person that his blood cells agglutinated with the anti-A, with the anti-B, and also with the anti-D, it means that he is AB positive. If no agglutination with the anti-A or anti-B nor anti-D, it means he is O negative. If there is agglutination with anti-A, no agglutination with anti-B, and agglutination with anti-D, it means the person has a blood type A positive, and so on. With this slide, I end my session. Thank you for your patience, and I hope you got some information or some useful information related to the blood groups, blood types, and their importance in the blood transfusion and the avoidance of blood transfusion reactions. Thank you very much.